Yeah, all mine are totally done. It's got what's yeah. If you want, I can run those tests down to the test. Okay, gotcha.
Then I'll be then all of them running.
So I do have.
Cool.
Remind me. Give you an opportunity to pass it out to anybody who wants one.
Yeah.
It's like the top.
Yeah, that should be fine.
If you know this circumference at the top.
We don't.
Good afternoon, everyone. Let's go ahead and begin class. Is somebody willing to volunteer to give us a prayer? Get us going. Please.
Heavenly Father, we're thankful that we're able to gather here today to learn about biochemistry. We pray that our minds can be quickened and that we can understand and understand the concepts that are being taught. And we pray that those in charge of making the vaccines can proceed with that. And we're thankful for all that we have. In Jesus' name, Jesus Christ.
Amen. Thank you very much. All right. So the exam 2 is being graded, and it's well along, but it is not completed as yet. But in the next couple days, hopefully, we will have those back in your boxes. Yes.
And the grades in the grade book are final. So if you see a grade, like we've done all the grading for those. So.
Okay, so I don't know if you've heard that, but if there is a grade in Learning Suite, that is the final grade. It's not a partial grade from one of the TAs who are grading the exams. But I don't know that any are back in the boxes as yet.
No.
So, no, not as yet. But shortly, we will have them back. Also, I do have an updated study guide for exam 3. I did post an older study guide, which is fairly quite close to what you will see for the updated one. I posted the updated one on Learning Suite, and I do have a hard copy for any in the room who would like to have that. So please raise your hands if you would. Okay. Looks like most would. So exam 3 will include the last couple items regarding glycogen regulation, synthesis and breakdown regulation. It will not include the pyruvate dehydrogenase complex. That will be on the final. As I mentioned early in the semester, probably the first class, the final is not comprehensive, but it is detailed in the same way that the other exams have been. It likely will have a somewhat different structure. It probably will not have, it will have more multiple choice questions. It will have several fill-in-the-blank questions, but it won't have the sort of longer questions at the end. That's the final. The current one does have 4 questions that are a little bit longer, more involved, and worth more points. Yes?
So what you're saying is like everything from exam 2 to now to before the pyruvate dehydrogenase complex is on exam 3, and then everything from pyruvate dehydrogenase complex to the end of the semester is the final.
Yes, that is correct. So, yes?
Will that be online or in the testing center?
There is no testing center availability for the final. So what I think will happen, this class has been given a day and time for the final, and I think we'll observe that. The exam will probably be administered through Learning Suite. You'll have a block of 3 or 4 hours, probably 4 hours in which to take the exam, but on a specified day at a specified time, at least to begin the exam. This is a little different, but the testing center was much more flexible, and I realize that can be a bit of a headache, but I don't really have many options given me, so yeah. Yes?
And so is the material today, like everything today on the final, or?
So from here up is on exam 3 because it still deals with glycogen regulation. The pyruvate dehydrogenase complex, citric acid cycle, and beyond will be on the final. So a little bit of today on exam 3, which officially starts tomorrow, and the rest on the final. Other questions before we forge ahead? All right, let me begin. I want to consider so let me take you back. Okay. All right, I want to go back to the end of the last class. We were talking about the process by which glycogen synthase was inactivated. And again, this is under hormonal control, but the actual mechanisms are not known. I mean, the enzymes that carry out the inactivation are known, and we talked about those last time, but how those enzymes are regulated is unknown. So it leaves a little bit of a mystery. We talked about, let me go back one. We talked about this process. So if you look at the upper part of the sort of circle there, we talked about this process in which we phosphorylate the glycogen synthase. And the first enzyme that adds a phosphate is this casein kinase 2. And this is considered to be a priming event and not a complete inactivation. So it adds a phosphate, and that phosphate is necessary for the binding of glycogen synthase kinase 3, the GSK3, which is going to add several additional phosphates to the glycogen synthase and inactivate it. So that's what we talked about last time. Now, we had talked about this sort of remarkable process in which the GSK3 actually moves along the glycogen synthase, adding phosphate after phosphate after phosphate after phosphate to serines that are spaced 4 amino acids apart. So the GSK3 has this array, this available piece on it, which is embedded then in the GSK3, phosphorylated in a serial fashion, and in doing so is inactivated. You'll recall that this first phosphate, the priming phosphate, has a specific binding site on GSK3. It has all these positive groups in that cavity. Those will surround and be bound to the initial phosphate. But then, as we have the catalytic site over here next door, where we add a phosphate from ATP to an available serine. Once this happens, then the entire GSK3 moves, and the binding site now overlays this newly phosphorylated serine. We place the catalytic site over an available hydroxyl group from a serine. We bring in another copy of ATP, and the process continues. It turns out that the GSK3 is itself regulated. Everything seems to be regulated, and sometimes in different ways, which makes it hard to keep track of, admittedly. But in this case, the GSK3 is regulated at least in one direction, at least in its inactivation, under the control of insulin. So insulin will bind to its receptor. That's what's shown here in this sort of upper left-hand corner. It binds here, and it leads to the activation of a protein kinase B. Just to make things confusing, so protein kinase A was under the control of glucagon, but we now have this additional kinase to keep track of, this PKB, which is activated by insulin binding to its receptor and some downstream events from that process. This PKB phosphorylates and inactivates GSK3. So let's walk through the logic behind this. If insulin is up, that's an indication that we have adequate, even abundant carbohydrate in our bodies, increased glucose in the circulation, increased glucose in the cell, and so we would, if we were going to do something with glycogen, we would want to synthesize glycogen, and we would want to inhibit the breakdown of glycogen. Okay. We know that insulin leads to the activation of PB1, which will activate all of the phosphorylated copies of GSK3. But in this step, what happens with PKB is that we're going to phosphorylate and inactivate the enzyme that inactivate the enzyme that inactivates glycogen synthase. So PB1 will dephosphorylate and activate all of the phosphorylated GSK3, glycogen synthase. Insulin will also lead to the phosphorylation and inactivation of the GSK3, which means that all of the activated glycogen synthase will remain active. It will not be phosphorylated by GSK3 and inactivated. Okay. Complicated. All right. this inactivation of GSK3 by phosphorylation by PKB is interesting in that it also is going to phosphorylate a serine on a flexible piece of the GSK3 that can fold back on itself and fit into the binding site, the substrate binding site of GSK3. So the site that would normally be occupied by the phosphate added by casein kinase 2 is now occupied by the phosphate added by PKB. So there is no way that it can bind to the glycogen synthase. So it prevents then its binding and its phosphorylation of glycogen synthase. Now, what is not specified and what is, so far as I know, unknown is how do you dephosphorylate the GSK3? We understand how it is phosphorylated, this PKB under insulin control, but we have no mechanism for its dephosphorylation and reactivation, but it does occur. So it occurs, but through mechanisms that have yet to be identified. Okay, let me give you one more concept. This one will be lightly touched on in the exam. You should know at least what this GM, okay, so you'll notice this GM over here. This is called the glycogen targeting protein. And what this is showing is that the glycogen synthase, the glycogen phosphorylase, okay, glycogen synthase, the enzyme that synthesizes glycogen, and glycogen phosphorylase, the enzyme that breaks down glycogen, and the phosphorylase kinase, which is the enzyme that activates the glycogen phosphorylase, all exist as a complex. So all three of these enzymes associate or are one with another in a complex that sits on the surface of the glycogen particle itself. So this massive granule that is made up of these many, many branches of glycogen actually have these protein complexes on the surface available for activity. Now, we have both the synthetic and the degradative pathways represented in this complex. The glycogen targeting protein is responsible for bringing the phosphoprotein phosphatase 1 to the granule, to this complex on the granule. So to do that, the glycogen targeting protein needs to be phosphorylated with just a single phosphate. So as it's phosphorylated by protein kinase B under insulin's direction, okay, again, so here's PKB, a second instance where it is involved in the glycogen regulation. It's going to monophosphorylate the glycogen targeting protein, which now will bind PP1 and bring it to this protein complex. You'll recall that the PP1, the phosphoprotein phosphatase 1, will dephosphorylate all three of those enzymes. The three enzymes being the glycogen synthase, which activates it. It's going to dephosphorylate the glycogen phosphorylase, which inactivates it, and it's going to also dephosphorylate the phosphorylase kinase, which inactivates it. Consequently, increased insulin in response to higher blood glucose levels then is going to promote glycolysis by phosphorylating or activating the PKB, phosphorylating the glycogen targeting protein, bringing PP1 to this protein complex on the glycogen granule and leading to the activation of glycogen synthase, which is going to then increase the amount of glycogen stored. You can see over here that if we become hypoglycemic, epinephrine and glucagon are those two hormones that are two of the four hormones that are increased in response to hypoglycemia. And epinephrine will bind to its cell surface receptor, or glucagon will bind to its cell surface receptor in liver cells. And this leads to the activation of this protein kinase A. We've seen protein kinase A in several settings related both to glucose regulation and glycogen regulation. What happens then is if pKa is activated, it adds now a second phosphate to the glycogen targeting protein. This leads to a dissociation of the PP1 from this complex that contains the glycogen synthase and glycogen phosphorylates. And that PP1 is then, it also leads to the phosphorylation of an inhibitor protein that will interact with, bind to the PP1 and prevent it from dephosphorylating these enzymes. In addition, the glycogen targeting protein now dissociates from the complex. It goes off and is, in effect, is not able now to bring PP1 to the complex further. What is not known is how the glycogen targeting protein is dephosphorylated to allow for this process to recycle. And it is not clear what is going to dephosphorylate this inhibitor protein that is complex with and inactivating the PP1. Those processes are yet to be discovered. So that's where we are with the science. Okay, as I said, this will be lightly touched on in the exam. You should know about the existence of the glycogen targeting protein and that it is responsible for bringing the phosphoprotein phosphatase 1 to this protein complex, allowing the PP1 to dephosphorylate those three different enzymes that are part of that complex. All right, any questions about this? This is a very busy, complicated slide. I recognize that, and as I said, you won't be expected to draw this out or know the intimate details of this, but you should be aware of the glycogen targeting protein. Questions? Let me make one final comment with regards to glycogen. So let me pose the question to you. Why make glycogen? Why not just store glucose inside the cell? It would seem so much simpler at one level if you just had the individual copies of glucose or glucose 6-phosphate or something that could be immediately used, rather than having this complicated system of formation and breakdown of glycogen. So why is it that the cell stores glucose as glycogen? Any ideas? Yes?
Just because it doesn't need it.
Because it doesn't need it, okay, perhaps, okay, but if it doesn't, I mean, in one sense, if you didn't need the glycogen, you wouldn't need to store the individual copies of glucose either. So, I mean, you know, it's just having glucose available at some point when intake is low is going to be important. Any other ideas? Okay, I want to take you all the way back to freshman chemistry and talk about osmolality. You recall, you won't recall, maybe you recall, I don't know, this concept of osmolality that there is a pressure that is proportional to the number of particles that are present in a compartment. And these compartments are separated by semi-permeable membranes. And so you can actually develop an osmotic pressure if you have more of a salt or more of glucose dissolved in a solution on one side of this semi-permeable barrier. Well, the same is true in the cell. If you had individual copies of glucose, so let's say a glycogen particle has 50,000 copies of glucose, you would have 50,000 times the osmotic pressure inside the cell. And this would lead to the rupture of all cells in the body. So the cell is pretty smart in that it wants to have glucose available. It needs to have glucose available during times of low intake. On the other hand, it is protecting itself because one massive glycogen particle exerts the same amount of osmotic pressure as a single copy of glucose. Doesn't make sense, but that is the reality. Okay. All right. We are leaving now the material for exam 3, and we are now moving on to the final material for the semester. This idea of respiration, this idea of oxygen consumption, this idea of taking materials that have potential energy, chemical energy, and converting them in ultimately to CO2 and water happens primarily in the mitochondria. So this mitochondria is a specialized organelle. It is the energy center of any given cell. And when we talk about energy formation, ATP formation, all of those processes, they are housed in the mitochondrial matrix. Up till now, glycolysis and the majority of gluconeogenesis has taken place in the cytosol. But now we are moving into this specialized compartment within the cell, and we're going to talk about processes that happen there. Another concept is that the body is able to be very economical. It's able to take protein, fats, and carbohydrates and convert them into the same common chemical species that is going to feed into the citric acid cycle, the Krebs cycle, the tricarboxylic acid cycle. And this is where we're going to experience, or these molecules are going to experience multiple oxidations. The energy released as part of those oxidations is captured by NAD plus in its reduced form, NADH, and by FAD in its reduced form, FADH2. The molecule that that is the common, if you will, common chemical species is this acetyl-CoA. It's an acetate coupled to a long molecule called coenzyme A. We'll see coenzyme A here in a minute, but the coenzyme A then is bound to this acetate, this 2-carbon piece, which is the remnant of glycolysis or fatty acid breakdown or protein breakdown, is going to then be oxidized. It's going to be oxidized. There are going to be a series of 4 oxidations in the citric acid cycle per 2-carbon segment that enters as acetate. Okay? Right. Today we're just going to focus on now how the pyruvate, which is the end product of glycolysis, is going to be converted into acetyl-CoA. And you'd think it would be a fairly straightforward simple process, but it turns out to be very complicated. I guess that shouldn't surprise us at this point. Let me just reemphasize. Here is just in outline the citric acid cycle. You can see at the very top, we have the acetyl-CoA, this common sort of denominator for all of these metabolic pathways that moves into the citric acid cycle. You can see that there are 4 oxidation steps. These are highlighted by the formation of NADH in 3 out of the 4 oxidations, a copy of FADH2, and we're going to produce a copy of GTP, which is energetically equivalent to ATP. So we get a lot of mileage out of one 2-carbon piece that enters this pathway. The other thing to keep in mind is not only is the citric acid cycle this oxidative hub, where we carry out multiple oxidations, it is also referred to as a metabolic hub, because those intermediates in the citric acid cycle are used for the synthesis of other compounds in the body. And that is what this slide is meant to show. So in the center here is the citric acid cycle, but you can see that we form purines, which are part of RNA, DNA, your porphyrin rings, pyrimidines, which again are part of the nucleic acids. And we have a series of amino acids that can be produced from intermediates in the citric acid cycle. So this citric acid cycle, again, is a representation of economy within the body. It has these 2 rather distinct functions. One of carrying out these oxidations to generate high-energy molecules, which lead to ATP production. At the same time, you can access those intermediates for synthetic needs within the organism. Okay, let me introduce you now to the pyruvate dehydrogenase complex. This is an extraordinarily large complex made of multiple copies of 3 proteins. And we're just going to refer to them, they have formal names, and we're just going to refer to them as E1, E2, and E3. And this is the organization of the pyruvate dehydrogenase complex. This is a cutaway of it. The yellow outer coating is all E1. Okay, this is enzyme number 1, and it's going to have access to the liquid inside the mitochondrial matrix. This is where the pyruvate is going to be. So the pyruvate is able to access E1. E2 and E3 form this core, and they're sort of interdigitated one with another, and there are reasons for that. E2 binds, this protein binds a special molecule called lipoic acid. It's going to bind it with a lysine, so it has a long, flexible arm, long enough that this piece that is attached to E2 can access the catalytic site of E1. It can also access a catalytic site on itself, E2, and it can access a catalytic site on E3. So this long, flexible arm acts a little bit like a derrick that's able to go pick up one thing and deposit it somewhere else, and then go off and be acted upon by E3. Let me go back. So there are other proteins associated with this complex, but for our purposes, we're just going to focus on the E1, E2, and E3. In bacteria, there are 24 copies of E1, 24 of E2, and 12 of E3. In mammalian cells, at least in cows, there are 30 copies of V1, 60 copies of V2, and 12 copies of V3. Why the difference? I really don't know, but it means that there are multiple sites where these individual processes can take place. So each copy of V1 is able to carry out those steps associated with it. There are some special molecules that are associated with part of the catalytic cycles represented by the pyruvate dehydrogenase complex. These include something called thiamine pyrophosphate. This is a derivative of a vitamin B1. So thiamine B1 is something we have to take in. We don't synthesize this compound. So all of these cycles depend on molecules that are derived from the diet. If we don't have enough of our vitamins, then it leads to disease, and sometimes very significant disease. I talked about lipoic acid. This is a component. This forms this flexible arm attached to E2 that can access E1, E3, and a catalytic site on E2 as well. Okay. So, what do you need to know? You should know, you should be able to identify and describe, at least in words, not necessarily, you don't have to draw out the compounds, you don't have to do electron pushing, but you should be able to identify what happens at each of these 5 steps. Okay, and I'm going to walk you through this. I'm going to do a little bit of electron pushing at the beginning, but this is for your information, not something you'll be expected to know on the exam. So the first thing that happens is pyruvate binds to a catalytic site on E1. And it's going to bind not to the protein directly, but to this thiamine pyrophosphate. So if we look up here, let me just direct your gaze up here. This is, here we come in, here's pyruvate coming in. It binds to this TPP. Notice that the TPP is tethered to E1. And after the pyruvate binds, it's going to undergo a decarboxylation. So the CO2 minus is going to come off, leaving a pair of electrons. That's what's shown here is this has lost the carboxylate. This is formally an aldehyde now. It doesn't look like it. It looks like a hydroxyl group. It is, but if you were to decouple this from TBP, it would be acid aldehyde, acetaldehyde. This is going to form a carbanion, and it's going to attack this sulfur, which is part of the lipoic arm. So the very terminus of the lipoic acid, is this curious ring that has a disulfide bridge in its oxidized form. When you have this acetaldehyde attack the lipoic ring, that is an oxidation reduction reaction. The ring is reduced. The acetaldehyde becomes acetate. So it is formally oxidized at that point. This acetate then, coupled to the lipoate, which is tethered to E2, is going to access a catalytic site on itself. So this arm is going to swing over, and there's a site on E2 where we have this coenzyme A attached. There's going to be a transfer, a direct transfer from the sulfur of the lipoate to the sulfur of the coenzyme A. It's an energy-free process. It happens rapidly, and we have now formed acetyl-CoA, which comes off of E2. The issue then is that we need to reoxidize the lipoic ring to be able to receive another copy, another 2-carbon copy from the breakdown of pyruvate. So that happens on E3. Over here we have bound to E3 in a permanent way, embedded in it, is this FAD. It can't go out or come in. It is there from the outset of its being part of this complex. But those 2 hydrogens on the lipoic arm with 1 electron each are going to be transferred to the FAD to form FADH2. So that would be step 4 now. But because the FADH2 cannot dissociate from the complex, to be able to allow for another round of catalysis, we need to reoxidize the FADH2 back to FAD. So this is its catalytically active form. That is accomplished by a copy of NAD plus coming in, binding near the FADH2 and receiving the electrons from it, reoxidizing the FAD, H2 to FAD, and the NADH carries those electrons away and feeds into the electron transport chain. Okay, I only have a couple minutes here, but let me outline the first step or two of this. I'm going to do this on the board. So let me. Okay. I haven't. Let me show you TPP. This is going to make more sense if we understand. So here is TPP. Okay, crazy molecule. Okay, but the critical feature of this, at least in terms of the catalytic activity, is this ring, this heterocycle that has a nitrogen, a sulfur, all part of the ring. This hydrogen here is very acidic on this carbon, and it's going to easily form a carbanion, which is going to attack the carbonyl of pyruvate. Are you seeing the board? All right, so I'll do this relatively quickly because I'm not expecting you to draw this out, but I would like you to understand what's going on. So let me bring in here our pyruvate. It's present in, there is a transport system that allows for its movement from the cytosol, where it is produced as the end product of glycolysis, into the mitochondrial matrix where we have the pyruvate dehydrogenase complex. And here we now have this thiazolium ring that has all this stuff hanging off of it. So this is the thiamine pyrophosphate, but this is the working part of it. And as I mentioned, there's a proton here that is easily dissociated off, forming this carbanion, which is going to then attack the carbonyl of the pyruvate. This will push electrons out on the oxygen. It will pick up a proton from some feature on the enzyme. So this is attached. This thiamine pyrophosphate is covalently attached to E1, and these features here are also part of E1. So after this, I end up with not going to draw in all of the features here, but we now, now have covalently coupled pyruvate to TPP, which in turn is tethered to E1. So this is happening on E1. There's a catalytic site on E1. The enzyme has the ability to bring about the decarboxylation of this molecule. So as part of the catalytic process, we're going to lose this CO2, and this will leave us with a carbanion, pair of electrons here, on this species. This electron density is redistributed over these different nuclei. It's stabilized by the availability of the of these other nuclei in the unsaturation here. At this point, this arm is now going to interact with this flexible lipoid. So this feature that is tethered to E2 comes in and presents this ring, which is tethered now down here to E2. So this is tethered here to E1. It's going to come in, and this pair of electrons is going to attack this sulfur. This is going to come out and pick up a proton, and this constitutes an oxidation of this species and a reduction of this species. So this is step 2, where we actually have this interface between the TPP on E1 and the lipoate on E2. This opens this ring and couples now this 2-carbon piece, now formally acetate, to the sulfur of the lipoate. Then this base that was up here will re-abstract the hydrogen that was here. We will reform the carbonyl and we'll bring the electrons back onto the thiazolium ring on the carbon of that ring, which is stabilized again by this sort of unsaturated system. At this point, we have now coupled acetate to lipoate. Okay, I'm going to stop here. We'll come back and talk a little bit more about the subsequent steps. They go pretty quickly and there's not a lot of electron pushing, but this represents steps 1 and 2 of the pyruvate dehydrogenase complex cycle. Okay, questions? All right, then let me share a story. When I was an undergraduate, I was a chemistry major. I can't remember, I think it was during freshman chemistry. I had a chemistry lab. I don't remember how many students were in the chemistry lab. It didn't seem like it was overcrowded, but we were divided into pairs, and I had a lab mate that I did experiments with. A member of the class, there was a fellow there that I knew vaguely from other connections, and it was kind of interesting. During a lab class, especially as we approached submitting our lab books, our results for grading, he would sort of drift around the room. He would come up to us and say, I was just curious what you got. I just want to make sure I'm sort of in the same ballpark as you are. What did you get for, for this or for that? It seemed very innocent, and we would sort of say, well, this is kind of what we came up with, and, we're not really confident about our results, but That's what it was, you know, and I didn't think too much about it, but I began to notice that he didn't come just to us, but he, you know, a few minutes later, I would see him across the way talking to someone else at their lab bench, kind of looked like he was asking them about their lab results. Well, after a while, it finally dawned on me that this young man was not doing the lab experiments at all. He was dry labbing these. He was getting all of the necessary information needed from everybody else in the class to be able to submit a report and get credit for it. I didn't think much about it. I mean, I moved away from Utah, as I mentioned, and was gone a long, long time. But I began to collaborate with. Well, I came back to BYU. I came back to Utah and began working here at BYU, and I was surprised to actually pick up a newspaper and see the name of this young man. It turns out that he had gone to medical school, had become an obstetrician, and he was being debarred by the medical association here, and was being sued by many, many, many women for malpractice. I happened to be doing research in complications of pregnancy, and I had a close colleague, a collaborator up at the University of Utah, and I just said, you know, do you know this guy? And he said, oh yeah. I said, what's the deal? And he said, well, this guy always cut corners. He's got himself into a lot of trouble. And I thought, oh my goodness, you know, this has been something he has done all his life. He has not done his own work. He has not given it his best shot. He has sort of fabricated data. He's cheated the system, but finally he has been caught, and this is going to result in disaster for him and for his family, as they have to pay for the legal costs and will undoubtedly lose many of these lawsuits, which will cost him a fortune. Anyway, I simply state this, that I know that there are times when it is tempting to cut corners. Sometimes we don't have to put in our very best work. If you're going to wash dishes, it's good to do them reasonably well, but they don't have to be perfect. On the other hand, if you are expected to do your own work and being assessed on your own work, then I would encourage you to do the very best that you can, and be satisfied that you were honest and had integrity about it. Anyway, something to think about. Thank you very much. Good luck on the exam if you're taking it early, which I doubt. But